Thank you. So I'd like to thank the Equalize organizers for this opportunity and also my mentor, Dr. Jay Littker from Avalon Ventures. So I'll be talking about a novel therapeutic that we have identified, namely the kisspeptin receptor, or KIS1R, for the treatment of non-alcoholic fatty liver disease, also known as NAFLD. So our company's mission is to identify novel therapeutics to treat NAFLD, and to that end, we have unique insights how activation of the kisspeptin receptor is such a therapeutic. We'd like to take a dual approach to bring this technology to market. We'd like to conduct rapid early proof of concept studies in humans using TAC448, which is an injectable analog of kisspeptin. Kisspeptin is the endogenous ligand for the kisspeptin receptor. And in parallel, we'd like to develop an oral molecule by conducting a small molecule screen to identify novel activators of the kisspeptin receptor. We're seeking $35 to $40 million investment for the development of these therapeutics. However, our initial ask is $15 million for years 1 to 3. So there are no approved pharmaceuticals to treat NAFLD, and globally over 1.5 billion people have NAFLD, and in the US, 100 million adults and children have this disease, and our annual healthcare cost is over $103 billion. And so we actually have a method of use patent filed for the use of TAC448 to treat NAFLD and NASH. So NAFLD is a spectrum of diseases. The healthy liver does not store fat. That is the job of the adipose tissue. However, when there's obesity or dyslipidemia, the extra fat that's circulating in the body gets stored in the liver. And this is known as fatty liver or steatosis. And it shows up as these white vacuoles in the histology section. And I'd like you to keep this in mind because I'm going to refer to this later. So about 100 million Americans have fatty liver, although most do not know that. So these fat deposits cause the liver to get enlarged, and then the liver gets inflamed, and the liver cells known as hepatocytes get injured. And this condition is known as non-alcoholic steatohepatitis, or NASH. And as the disease progresses further, the patients can develop fibrosis or cirrhosis, and the liver gets scarred and is unable to function. And in fact, NASH is the leading cause of liver transplantation in the US right now. Some patients will actually develop hepatocellular carcinoma, or HCC, which is the fastest rising cancer-related deaths in the US. So the market is huge, and in the next 8 years, it's expected that there'll be $25.6 billion in total sales from drugs to treat NAFLD and NASH. The majority of these will remain inhibitors of fat accumulation. So the kisspeptin receptor is a G protein coupled receptor that when you bind kisspeptin endogenously, that's the agonist. And in doing so, it causes an increase in intracellular calcium level. So therefore, this can be used as a readout of receptor activation. And the liver expresses the kisspeptin receptor and actually produces kisspeptins. So TAC448 is a potent kisspeptin analog. It has a long half-life compared to endogenous kisspeptin. And this compound was actually developed by Takeda, who owns the composition of matter patent, which will expire in 2028. And Takeda has done phase 1 clinical trials with healthy men, about 82 healthy men, and also men with prostate cancer. And also they did phase 2 clinical trials with men with hypogonadism. And Takeda, TAC448 is now actually exclusively licensed to Myovant, who is using this drug for treating women's reproductive disorders, including infertility. So what's important to note is that the safety and the pharmacokinetics data is available and actually has been published in peer-reviewed journals. So we used a preclinical mouse model of NAFLD, where mice are fed a high-fat Western diet, and we used TAC448 as proof of concept study. So the liver of the control mice, as you can see, exhibits steatosis or fatty liver. And you can actually mark these lipids using a stain known as oil red O, which stains them red. So what we observed was that the animals that were treated with TAC448 for a month were protected from development of steatosis. And what we found was that the animals treated with TAC448, there was a reduction in hepatic lipids, which is a hallmark of fatty liver, reduction in serum ALT levels, which is a clinical biomarker for NAFLD and NASH, reduction in inflammatory and fibrosis markers, which is a hallmark of NASH, and they had lower blood levels of triglycerides, free fatty acids, and cholesterol. And what's incredible is we have brand new data showing that TAC-448 treatment actually improves fibrosis in mouse models. I just got this data, so I wasn't able to include it. So we now also have a mechanism of action. What we found was that TAC-448 treatment decreases the expression and activity of key regulators of hepatic fat synthesis, in addition to promoting fat breakdown. And we also have human data using human NASH liver biopsies for clinical relevance. So therefore, we'd like to take a two-pronged approach. We would like to perform a small clinical trial with TAC-448, which is a subcutaneous injectable drug as proof of concept. And in parallel, we'd like to do a small molecule screen to identify new activators of KIS1R to develop into an oral drug. So as I mentioned before, TAC-448 has been used in phase 1 and phase 2 clinical trials, and the dosing and toxicity data has been published. So we would like to therefore conduct a 6-month study where we'll administer 2 doses of TAC, a high dose and a low dose, as well as the placebo group. And the dosing is going to be a 2-week depot formulation based on the publication by Takeda with the phase 1 clinical trials. We plan to enroll 30 patients in total, 10 per group, and the study endpoints will include safety, serology, where various biomarkers will be measured every 2 weeks. Then we'll do monthly FibroScan to look at changes in liver fibrosis and steatosis, and then liver biopsy will be done at the start of the study and at the end of the study. And we expect to recruit 1 to 2 patients per week at the hospital where this will be done. And we're also exploring a possible collaboration with Myovant to actually get the cGMP grade drug and their clinical data. In parallel, working with a CRO like Genesis, we'd like to conduct a small molecule screen to identify novel activators of KIS1R to then develop into an oral drug. And this plan was actually developed in consultation with Genesis. So to start with, we will use 5.5 million compounds using a human cell line that my lab has developed and has published with. So we'll screen these compounds and using this cell line. And we will use the Flipper assay, which is a calcium assay, to look at the readout. And these 5.5 million compounds are also off patent. So then the top 100 or so compounds will be used in the primary assay. And then we will also test them using mouse hepatic cell lines. And we will also look at toxicity and specificity and their selectivity for activating KIS1R against other GPCRs. So then the top 5 to 10 compounds will be used to make sure that they can activate kisspeptin receptor in vivo using plasma luteinizing hormone as a readout. And then the top 2 compounds will be tested for their ability to actually work, the therapeutic abilities using the NASH model. So as I explained, we're looking to conduct phase 2 clinical trials with TAC-448 because this drug has gone through clinical trials with the same injectable composition and the safety data is published. And thus with financing, we will file an IND and conduct phase 2 in the second year. And then for the second track, we expect to have the lead compound within 3 years. The small molecule screen will take about $2 to $3 million. The lead optimization and IP filing will take 7 to 8 million. We'll need another 5 million to scale up and to do the IND enabling studies and IND filing. And finally, 15 million to conduct phase 1 and phase 2 trials. And for both tracks, we are seeking partners and licensing opportunities. So our technology, as I mentioned, there are no FDA-approved treatments available, but we believe our technology will have improved selectivity compared to those that are in phase 3 clinical trial right now, because this has actually been verified using knockout mouse models. And we also have a novel mechanism of action. So my team includes the director of hepatology. He is the one who also helped me design the clinical trial, Dr. Rutsky. I'm also working with endocrinologist Ankit Shah and Fred Wandersford, and a pathologist, He Wang. I myself am a cell biologist. I've worked with Kisspeptin for over 15 years, and I'm also working with another Kisspeptin specialist, Andy Batwa. So to summarize, there's a high unmet need, and the clinical trials with TAC-448 will be a short path to market for an injectable drug. In parallel, we do want to develop an oral molecule. And so I'm just summarizing what I just showed you. So the first request is going to be for 15 million for the first 3 years, which will give us phase 2 data with the TAC-448 compound from the first track, and will allow us to conduct a small molecule screening and the lead optimization studies from the second track. The second tranche is $5 million to do the IND enabling studies from the drug stream. And finally, 15 million for phase 1 and phase 2 trials with the new oral drug. And with that, I'm happy to take questions. And I'd just like to say a special thank you to Dr. Jay Littker for his mentorship. Thank you.
That was terrific. Judges, let's go ahead with any questions that you may have. And if anyone wants to, judges, just choose who wants to go first. Otherwise, I'll call.
Moshmi, I have a question about the expression of the KISS-IR receptor in other tissues. Is it expressed beyond the liver?
That's correct. So what we have shown, there is active expression in other tissues as well. In the liver, it's typically low, but what we found using human data is that the expression actually increases under fatty liver conditions. So we believe it's a compensatory effect that the body's having, but it's obviously not sufficient enough to safeguard against disease progression. And that is why we believe enhancing the activation is going to be beneficial. The expression is not ubiquitous. It is still relatively limited peripherally, how it's expressed.
Thanks.
And I have a question too. There was a great presentation. Thank you so much. It's kind of nice that you have TAC-448 as sort of a proof of concept. Just wondering if you understand yet whether or not that high and low dose is going to be sufficient in a different disease, given they've taken it forward in a couple of different diseases. That just wondering if you've done those animal studies, and in the animal study that you showed us, was that in a therapeutic mode or was that in a prophylactic mode?
Correct. So it was in a therapeutic mode. And so the dosing is something that, in mouse models, there are various doses that were tested, but, mouse and humans are going to be two different things. So, but what we found, and we actually had conversation with Myovant a couple of weeks ago, and they actually are interested in talking to us more about this. And they actually said that they have more clinical data that they can give us. But even if that did not work out, the data that Takeda has published based on their phase 1 clinical trials is going to give us a good starting point for us to try the high dose and the low dose.
Yeah, question, because your lead program is right now tied up with Myovant. You mentioned you're in discussions. And you said the IP expires in 2028. What are their plans? Do they have method of use that extends beyond that for their applications? And just curious, what level of signal? Because typically, right, they could stop you from developing their proprietary compound in the next several years. So, how far along are you in negotiating that? And then I have a question about the oral formulation as well.
Sure. So honestly, we're very early. So we just met with them a couple of weeks ago. The reason I waited is also I was waiting to see what happens with the fibrosis data in our mouse models. And now that, we just got the fibrosis data last week, that's why we were waiting to get that data before we proceeded further, because fatty liver is awesome, but fibrosis is actually, where people are dying. So to answer your question with respect to how will, how long will their patent cover, that information I do not have as yet.
Yeah, and you use descriptive terms to describe the preclinical data, and this is a well-trodden path. These fatty liver and obesity mouse models, there's different induction models. So I would encourage you to kind of show more of that data than use descriptive terms. But on the oral, what level of medicinal chemistry input have you gotten at this stage? Because presumably other folks would have tried to turn this IV into an oral, and how confident is your level in being able to create new IP around, right, the chemistry, and that it's achievable to get the drug-like characteristics you're aiming for?
So we're in the process of revising a manuscript that's going to be hopefully published this year in Journal of Clinical Investigation. And so our study is actually the first to look at this pathway in fatty liver. There is no, to the best of my knowledge, there is no other study out there. And so we engaged with the CRO for this reason, to remove bias. And they have a lot of experience working with GPCRs. They have a very high turnaround with respect to being able to, come up with, good drug screens. And then also all the compounds that they're trying are off patent. So, if the screen works, which I'm hoping it should, based on their success rate with other GPCRs, then, the turnaround should be very good.
Okay. Thank you.
Thank you.